Now with this background of information, let's continue where we left last time. So we were talking about the bacterial toxins. Am I correct, folks? We talked about the. Andrea, you had a question?
How many questions are on the
Extra credit quiz, maximally 5 questions.
Is it multiple choice or
That's like the regular quiz questions. So the bacteria, we talked about the toxins, we talked about some of the terms associated with the bacterial toxins. We talked about the fact why bacteria secretes toxin, location, the location of the genes for the toxin. It could be chromosomally coded. In certain instances, it's the phage DNA that codes for the toxin, and those phages, once it integrates as a prophage, that's when the toxin is basically expressed, And as I go along, I will let you know more, because I did talk about some of those phages which codes are the diphtheria, the cholera, the botulinum, and among the bacteria is the scarlet fever, of the streptococcus. Those are all encoded by the phage gene. Okay, and the plasmid, you know about the anthrax toxin is encoded by the plasmid. The chromosome 1, I think I did mention about the sugar toxin. Thank you. So these are some of the location of the toxin genes. Now what we are going to talk about is, and we also discussed some of the differences between the endotoxin and the exotoxin. And remember folks, this is not a comprehensive difference, but just something as a jump start for you all. These are a few of those differences between the exotoxin and the endotoxin. And lastly, we still wanted to start with the exotoxins. Most of the exotoxins are proteins. By nature, they are not lipid, they are not sugar, but they are protein, and they are secreted by many of those gram-positive as well as the gram-negative bacteria. And it's not bacteria is doing some extra work to secrete this toxin. It's a part of the normal growth and the metabolism. So it's a part of the normal growth and metabolism. Many of those toxins are recognized as foreign. So your body immune system gets activated and makes antibodies. So the immune system, it generates, so here the immune system generates antibody to neutralize the toxins. But the major problem what occurs, then why do we have all this problem? If your immune system recognizes the toxin, it generates the antibody, then why all this problem? The main problem is the damage by the toxin occurs much earlier before the antibodies are generated. So oftentimes the damage occurs before, oftentimes damage occurs before an adequate immune response gets kicked. But anytime you have the toxins in your body, they don't stay forever. Because after a while, the immune system will take over and it will neutralize those toxins. We got it, folks? But initially, you will get the rid of the toxin because initially the toxin goes, and before you get your immune system to kick in, the toxin has already done its job, but then the immune system gets kicked in, but then it doesn't prolong. The after effects or the effect of the toxin is not prolonged for a long period of time. Now, they are encoded by plasmid or the phage DNA. Most of the exotoxin, you can also write they're encoded by plasmid, phage DNA mainly, and occasionally chromosomal DNA. I think I should have. Oh Lord. It's all good. There are three types of these toxins. There are three types of the exotoxins, and those three types are based upon the tissue types they target, because most of the toxin they target some specific tissue, just like a hormone targets a specific cells of the body. By the way, folks, anybody who was absent who doesn't have these notes? Anybody? Thank you. There are some toxins which are grouped as cytotoxins. There are some which are grouped as enterotoxins. There are some which are grouped as neurotoxin, depending upon what is their target cells. The neurotoxins are the one that specifically target some of the neuronal cell or the nerve or cells of the nervous system. The enterotoxin, they target cells associated with the GI system or the gastrointestinal system or the digestive system. So to begin with, the cytotoxins, what they do is they either kill or they alter the functioning of the host cells. And this cytotoxin, there are many different types of cytotoxins. One is, a classical example is the pneumolysin. Pneumolysin is one of those exotoxin, and that pneumolysin, basically what it does, it is secreted by Streptococcus species, secreted by the Streptococcus species, or Streptococcus pneumonia to be more precise, by Streptococcus pneumonia. And what it does is it binds to the ciliated, to the cholesterol of the ciliated epithelial cells and make holes. So this pneumolysin, it binds to the cholesterol of the ciliated epithelial cells and makes whole. And one of the consequences when the holes are made in the cells, obviously after some time the cell dies. And the cell dies eventually. That's one aspect of the pneumolysin which belongs to the cytotoxin. It also inhibits some of the other things in the phagocytic process, but I don't want to make it more complicated. Just remember, pneumolysin is a cytotoxin made and secreted by Streptococcus pneumonia, and what it does, it makes whole by binding to some specific ciliated epithelial cell. It binds to the cholesterol of the membrane, and then it exerts its action. If you have not really been happy with this one, I'll give you one more example, and that is the diphtheria toxin made by the Clostridium diphtheria. The second one is the diphtheria toxin, Diphtheria, diphtheria toxin, which is secreted by the Corynebacterium, diphtheria. And what this bacteria does, the toxin which is made by this bacteria and secreted, oftentimes it targets the elongation factor 2. So this stops the protein synthesis. So this toxin, it stops protein synthesis of the target cell. And how does it do that? By inactivating eukaryotic elongation factor 2. This specific elongation factor 2 is involved during the translocation step of protein synthesis. So during protein synthesis, stop protein synthesis of the target cells by inactivating eukaryotic. So EIE, this E stands for eukaryotic elongation factor 2. Eukaryotic elongation factor 2, and this eukaryotic elongation factor 2 is responsible, this is responsible for the translocations, or this functions in the translocation step of protein synthesis. This functions in the there's something more to it, but again, I would like to keep it simple. What it does, it modifies the eukaryotic elongation factor 2. There's a chemical modification. It makes what is called as, it ribosylates the eukaryotic elongation factor 2. And that eukaryotic elongation factor 2 is the one which is involved during protein synthesis when the ribosome moves from one codon sequence to the next codon sequence. So during protein synthesis, this eukaryotic elongation factor 2 plays a very critical role, where the ribosome moves from one codon to the next codon due to the action of this eukaryotic elongation factor 2. And if this elongation factor 2 is modified, if it is chemically modified, then you stop the protein synthesis because there is no translocation. When the ribosome tries to move, this is the messenger RNA, and when the ribosome tries to move from one to the other one, it gets stopped because the eukaryotic elongation factors are no more functional. Because there is no protein synthesis, the cell eventually dies. The cell eventually dies because of no protein synthesis associated with those targeted cells. So that is the diphtheria toxin. If you're still not happy, I'll give you one more, and that is the anthrax toxin. The third one, I'm going to write it over here. The third one is the anthrax toxin, which is made by Bacillus anthracis. So the third one is the anthrax toxin, and that is being made by Bacillus anthracis. This anthrax toxin, I think I have the anthrax toxin more in detail, The anthrax toxin is here. We are going to talk about the anthrax toxin. This is the diphtheria toxin, and I'm going to talk a little bit more about the diphtheria toxin. And so these are the three of the toxins which are all belonging to the cytotoxins. There are a lot more, but let's try to catch it. Let's try to keep it simple. The pneumolysin is encoded by the chromosomal DNA. This is encoded by the chromosomal DNA. The diphtheria toxin is encoded by the phage DNA. And the last one, the anthrax toxin, is encoded by the plasmid DNA, as you all know. So the plasmid DNA is the one that carries the sequence or information for the anthrax toxin. with this background information, if still this has not excited you, there's one more exciting one, and those are the Shiga toxin or the Shiga-like toxin, and these are some of the very potent toxins which are made by some of those E. coli, the E. coli, like the hematogenic E. coli, like the E. coli 0157 that makes it up, as well as the Shigella bacteria. So they're both codes for the Shiga toxin. And Shiga toxin or the Shiga-like toxin is also a cytotoxin. Let's talk about it.
You said by Shigella and.
E. coli and some of those E. coli, some of the E. coli strains like the 015787.
So all the ones you talked about are the ones that kill,
All the ones that I've talked about are the?
are the ones that kill and affect the whole, right?
Yeah, they normally kill the target itself. The Shiga toxin is one of the very potent toxin which is made by some species of E. coli as well as the Shigella bacteria. E. coli is made by E. coli 015787 as well as the Shigella bacteria. It's called as Shigella because it was one of the Japanese scientists who first characterized this bacteria. His name was something first name and Shiga. So then we call it a Shigella bacteria.
And what does the Shigella toxin do to the body?
What the Shigella toxin does, there are two things what it does. And most of the toxins, when we talk about the cytotoxin, here is the A part, because most of the cytotoxins, or most toxins, put it this way, most of the exotoxins, they have two parts. There's a part which is called as the A part, and there's a part of the toxin which is called as the B part. The B part is the one that binds to the target cell. So B for binding that's the part of the toxin that recognizes the target cells, and it goes and binds on the surface of the target cell. Then the other part, the A part, is the one which enters inside the cell. A part is usually the active part. That is the active part, and this is the one that enters inside cells. Most of the time, this is the pathway they follow in a more general way, but although there are some deviations where sometimes both A and the B part, they enter inside the cell, and as we go along, we'll catch up more on this one when we talk about the cholera toxin and the diphtheria toxin in more particularly. So the A part is the one that enters inside some of those endothelial cells. The endothelial cells are the one that lines up the blood vessel. So it enters inside the endothelial cell. These are the one that lines the blood vessel, and specifically target the endothelial cell of some of the smaller blood vessels. It targets some of the endothelial cells of the smaller blood vessels, like the one that supplies the kidneys. And then it targets some, I said here reacts with ribosome because I don't want you to go with all the 28S and the 30S different types of proteins which are associated with it. So it reacts with the ribosome, and again, it halts the protein synthesis in the targeted endothelial cells. So it also stops the protein synthesis of the endothelial cells, and eventually when the protein synthesis is stopped, the cell dies off. this toxin or the Shigella toxin or the Shigella-like toxin is also responsible for another clinical condition which is called as hemolytic uremic syndrome. So this is also responsible for what is called as a hemolytic uremic syndrome. And what happens in hemolytic uremic syndrome, the red blood cell gets broken down in some of the tiniest, smaller blood vessels. Specifically, some of the smaller blood vessel that supplies to the kidney. So the red blood cell gets stuck on that one, then eventually they lyse, and then it results in kidney failure, anemia, because the red blood cells, they are dying, and there will be kidney failure, and that is accompanied by paralysis and some of the nervous injury signs. Why the nervous injury signs is associated with this Shiga toxin is because your nerve cell or the brain cell requires a continuous supply of oxygen. Am I correct, folks?
Yes.
So the brain requires a continuous supply of oxygen, and since the red blood cell, they take the oxygen from the lungs and then they go and supply the oxygen. Eventually, you will have a depletion of oxygen concentration. And once the oxygen concentration goes beyond a threshold, it falls below a threshold, then the person will kind of getting disoriented, and that's why the neuronal system injury sign, those keep on showing at a later time. One of the things I want to mention here is the Shiga toxin, which is encoded by the E. coli 015787, and this is a chromosomal DNA, is a chromosomal toxin. Remember, like when we had this problem with some of the fast food restaurant and the E. coli 0157 outbreak? Remember sometimes back, folks? I'm sure you all were born that time. What happened? Who were the biggest sufferers?
Jack in the Box, I think, was one of them.
Oh, possibly.
Chipotle, but kids got sick.
Who became most lethal to whom? Children's,
And all
elderly people, the immunocompromised individual, they were the most badly hit. Several children that died. Guess why? Most of the children's, elderly people who died eating those tainted beef, they all died because of kidney failure. Most of the children who died when they had that tainted beef, they died of kidney failure because of the hemolytic uremic syndrome associated with this toxin. Possibly, I'm not asking you to eat tainted beef, but possibly you can still bear with it. That's because your intestinal microflora is very healthy. And if you'd have some of the E. coli 0157 in your system, your body's normal microflora will outcompete it. But in children who do not have a full-blown intestinal microflora, they are more vulnerable. So are the elderly individual. Their intestinal microflora also kind of, it declines. So are the some of the immunocompromised individuals. So that's why the Shiga toxin becomes much more potent and it causes more harm to such groups of individuals. Does that make sense? Everybody's on the same page? So this is, can I move on? Yes, dear?
Oh, no, I'm stretching, sorry.
Oh, okay. So these are some of the cytotoxins. We talk about this one. The next slide, I think it talks a little bit about the diphtheria toxin, which you don't have to know everything in this one, but I'll let you know what exactly I want you to know.
What about
Oh, wait a minute, neurotoxin and.
I'm going to do that. I'm going to do that. Let me finish up the cytotoxin first. Just to get a generic idea, don't have to be very nitty-picky on this one. As I mentioned earlier, most of the cytotoxin, they are AB type of toxin or they are dimeric toxins. The B part binds to some specific receptor, and then there's an uptake of the A part inside the cell. In this instance, in the case of the diphtheria toxin, both the A and the B part gets inside the cell via a vesicle. And inside the vesicle, what happens is the inside of the vesicle becomes more acidic. And when the inside of the vesicle becomes more acidic, somehow that uncoupled the A and the B toxins, those are separated out. So that's when the A and the B subunit, they dissociate, and the A part moves inside the cell, and once it moves inside the cell, there's ADP ribosylation of the elongation factor 2. So now the eukaryotic elongation factor 2 is modified. It becomes ribosylated or ADP ribosylated elongation factor 2, and once it becomes ADP ribosylated elongation factor 2, it just cannot function anymore in the translocation process. The B part again is taken inside and is basically is tossed out from the targeted cells. You don't have to worry about the clathrin coated pit because this is not a cell biology class, but at least I want you to know that both the components are taken inside, and then in the endosome, once the pH gets lower in the acidic side, that's when it induces the dissociation of the A and the B subunit. The A subunit goes inside the cytoplasm, and once it is inside the cytoplasm, there is a modification. There is some modification, which is the ADP ribosylation of the eukaryotic elongation factor 2. And once that occurs, everything comes to a standstill. The protein synthesis is stopped because the elongation factor is a modified form.
And then fast forward, you're depleted of oxygen.
Here, what happened? The cell dies, and once the cell dies, the new cell will also die because of the toxin, and they all get clumped together, specifically in the upper respiratory tract, and eventually the person gets asphyxiated or they die because there is no oxygen cannot go through.
Okay.
You see? Yes, please.
Okay, so the A part kills the cell, the B part gets thrown out.
Yes, the A part, basically what it does, the A part, indirectly it modifies the eukaryotic elongation factor 2.
Right.
And once it is modified, it stops the protein synthesis because elongation factor is involved in protein synthesis. And the B part, it is basically is driven out from the cell. It just goes back because this endosomal vesicle, it will be basically going back. This part will become a part of the membrane, and the B part will be exocytosed to the exterior.
So then how does it travel then? How does it travel to another cell?
How does it travel to other cells? Is again, there are bacteria. Bacteria will be making more of the exotoxin.
Oh, okay, so when that B part gets thrown out.
That's it. It's done deal.
It's
You know,
done
it's
deal.
basically done
But
deal.
the AB, the dimetric, is continually making.
It's being continuously made by the bacterial cell is being secreted in the surrounding area.
Okay.
And if there are about 10 bacteria will continuously secrete the dimeric AB toxin, and that will keep on killing more and more cells. Oh, that also reminds me, folks, not now, let me finish up these ones. And then the last 10 minutes, I'm going to talk about the cell wall because I think there's a lot of confusion on the cell wall. I thought that everybody is clear, but there is some confusion, so I will try to clarify. Okay? Happy? Okay.
Thank you.
Yes.
Does it just take one dimeric exotoxin, one AD unit to kill the cell, or do you need several coming in?
No, just one. Just one will be good enough because once this ADP ribosylation occurs, by the toxin is like a catalytic, just like an enzyme. It will keep on
Okay.
modifying all the eukaryotic elongation factors. So it's
Okay.
more like a catalytic activity.
Okay.
So one cell, only one is good enough. Okay? Whatever. Are you okay? Are you happy? So this is the part, forget about this part. Folks, I want you, but this has become a little bit too much for this class, you know. So don't worry about this.
X right through it.
Huh? Yeah, you can just write here, don't worry about it. Don't cut it off like this on this colored picture. Now, and this part is not in your textbook, it's from a different textbook. I basically,
Stole it.
I borrowed it, I didn't steal it. But I'm still putting it here, permission required. I didn't take the permission. Now let's talk a little bit about the anthrax toxin. I think I'll take some time. You want to take a break, folks?
Yes.
Okay, get with your break, and then when you come back, we'll talk about the anthrax toxin, then the botulism, tetanus, cholera. Good job. So let's take about like a couple of minutes break, and then we come back and then we'll start. Yepsy.
I wanted to see if you brought the homeworks that we turned in. I forgot to highlight the correct answer, but I wrote them down.
Can you do me a favor?
Yeah.
When you come to the labs, I didn't bring it here.
Okay.
Huh?
All right, sounds good. Thank you.
Yes, Alan.
Are you going to make the announcement about giving us homework this weekend?
Which one?
Giving us homework too?
Yes, I'm going to do the homework. I better do it now. I will try to do it tonight only because it's otherwise getting late.
Silly question for you. I started out the class doing
That's okay.
Is that okay?
That's okay,
Is
but
it
later
okay?
on you did it. You
I
probably.
just started, so it's okay. I'm not gonna act for it. No. Okay. Okay. Thank you.
You have a clean writing also, so I'll enjoy.
A lot of labs, so I was worried. Weird, but you can see all the We're a few days apart.
He's like, Lord. You don't know. He asked me just like, please, please.
I've never seen that.
Thank you. No problem.
Surprising because every time we were working together, she came in and started doing it.
Really?
No, he was like,
It's the only source of caffeine that I'm really sensitive to.
You're addicted. Was everybody like that?
I think the lowest is 10. Head rush. That's me. No, okay, but I thought it I think on average, it was probably
Oh, yeah, that's it. Yeah, right.
that's frustrating. They might be disappointed. What is it? Get it out.
The only thing I know about nursing is that you burn. You can eat more food.
Yeah, I remember we were talking about just nursing, like being a nurse.
Oh, no, there was like breastfeeding.
This is my hair.
It's really taxing. I don't know if you knew that. Feel like butter.
I started studying today.
You've done a lot in your life.
It's like, it makes sense.
Oh, that's fine.
It's really like the labs are I'm not like
All you have to do is like
That made it work. I know. And it's gonna be plus 5. Okay. But I mean, like, more than one for sure. But 6, not 6. I like to you don't mess with it. I don't eat it. Oh, why? I don't like the tea. Oh, really? Are you big too? No. That's what I said. I'm gonna show you. I don't know. I'm like, I'm not bringing that home. It's hard to teach. I'm sure it's a nice but we moved in here. Oh, okay. I haven't even like looked into what I'm supposed to do. I'm not trying.
Oop.
Okay. Yeah, we are going to get in there. So, folks, let's continue with the cytotoxin. So, on the cytotoxin, one of the things what we talked about the cytotoxin is we talked about the pneumolysine, we talked about the diphtheria toxin, the third was the anthrax toxin. So, I'm going to talk a little bit more in detail about the anthrax toxin and what is its action. Basically, the anthrax toxin is a three-part toxin. There are three components to the toxin, and all the three are essential for the full action of the anthrax toxins. Now, the one part is called as a protective antigen. Protective antigen. Why it is called as a protective antigen, I'll tell you in a second. The other are the lethal factor, and the third one is the edema factor. So the lethal factor, protective antigen, and the edema factor are the three components of the anthrax toxin. For the anthrax toxin to be effective, you need all the three components. The one it is called as a protective antigen because earlier times the vaccine of the anthrax, they had this specific one. This was a component of the anthrax vaccine. That's why it was called as a protective antigen. And the lethal factor and the edema factor are the two other components of the toxin. And what the protective antigen does, the protective antigen forms a kind of a donut-shaped structure in the membrane of the target cells. So it forms a donut-shaped structure. So a protective antigen binds to one of the receptor. A small portion of it will get clipped, which is not so essential. What you should know is it forms a donut-shaped structure, and once that donut-shaped structure is formed with a hollow internal channel or a cavity, this is through that one. The other two, the lethal factor and the edema factor, is taken inside the cell. So the protective antigen forms sort of a transmembrane type of structure through which the lethal factor and the edema factor eventually gets inside the cell, and once they get inside the cell, there's a whole series of signaling and the pathway is activated, which eventually results in the cellular death. I just want you to know this much, that the protective antigen form goes and binds. It forms a donut-shaped structure on the cell membrane, which allows the entry of the other two components. And once those two components are taken inside, then those will now lead to some of those pathways which eventually lead to cell death. Not more than that, like don't worry about all the things over here. Although at one point I thought of talking about it, and I want to keep it straight and simple. Okay, so these are the three components of the anthrax toxin. Remember, anthrax causes, depending upon how it is, I think we talked about it earlier. Am I correct, folks? Huh? We talked about it earlier because there are three ways the disease manifests itself in three different ways: the cutaneous anthrax, the inhalation anthrax, and the gastrointestinal anthrax, depending on how you acquire the pathogen. Anthrax is a disease of cattle. It's a disease of herbivores animal, and that's the reason why veterinarians they are always vaccinated against anthrax. Zoo workers, people who work in the leather industry, they are all vaccinated against anthrax. And the general population is not given an anthrax vaccine. The general population, no. But some of these specific group of individuals like veterinarians, zoo workers or zookeepers, people who work in the leather industry, they are also vaccinated against anthrax. There is a push to get a better and improved version of the anthrax toxin. I'm not sure what step are we in, because I remember a few times back, somebody was working, there was a big push to develop a more potent and much more effective form of the anthrax vaccine. All right. I'm sorry, I don't know the status of that right now, but this is about the anthrax. So with all of this background of the cytotoxin, and I'm hoping you have got a little bit comfortable with the toxin part, or with the cytotoxin. Now let's talk about the neurotoxin. The neurotoxin is the one that impairs some of the action of the cells which are associated with the nervous system. And that's the reason why they're called as neurotoxin, because oftentimes they target the release of some of the neurotransmitters. Oftentimes they will impair the secretion of some of the neurotransmitters. Remind me at 7:30 to stop and go with the cell wall. Okay, folks? So, two of those most important amongst the neurotoxin includes the tetanus toxin, which is also called as AKA tetanospasmin. And the other one is the botulism toxin. Both of these toxins are secreted by the anaerobic bacteria called as Clostridium tetani or the Clostridium botulinum. So they both are secreted by the anaerobic Clostridium. Clostridium tetani or the Clostridium botulinum. So they are by Clostridium tetani or the Clostridium botulinum. The enterotoxins are the one that target the cells of the gastrointestinal system. And oftentimes, they cause some of the intestinal disturbances like a diarrhea, watery diarrhea, or the bloody diarrhea. And one of the classical examples of the enterotoxin is the cholera toxin. The cholera toxin is a classical example of this, of the enterotoxin. Okay? Now, to get on to the neurotoxin, let's talk about the tetanus toxin and what does it do and its action, which is shown over here. To get a good idea, I'm sorry, this is the botulinum toxin.
So sorry, you said that the cholera was
Enterotoxin. Cholera is an enterotoxin. Now, to get an action of the botulinum toxin and to appreciate the action of the botulinum toxin, I would like you to get a little idea at the neuromuscular junction. How is that process of, how the process at the neuromuscular junction occurs? A little basic aspect of that one is, at the neuromuscular junction, so the muscle cells are innervated by some of the nerves, and at that neuromuscular junction, you have what is called as the synaptic cleft. The synaptic cleft, and I'm sure you all know about it, but just in case if you have not taken anatomy, and correct me if I'm wrong on anything because my anatomy is very poor. So here you have the muscle plasma membrane, and here you have the nerve endings. The nerve endings have this membrane also. This is the neuronal membrane. And the space between these two is the synaptic cleft. And on the nerve ending, you have, at the ending, you'll have many of these small bag-like structure which are overfilled with the neurotransmitters. Okay? So there are some neurotransmitters which are excitatory, some of the neurotransmitter which belongs to the inhibitory neurotransmitter. One of those is the acetylcholine. So acetylcholine comes in at the neuromuscular junction, and this vesicle, the vesicle, it fuses with the membrane of the nerve plasma membrane, or the neuronal plasma membrane and the vesicle membrane, they fuse, and upon fusion there's exocytosis of the acetylcholine within the synaptic cleft. That acetylcholine which is released in the synaptic cleft will go and bind to some of the receptor of the acetylcholine on the membrane of the membrane of the muscle cells, and that's when it will bring about some of this action in the muscle cell. This is the normal, straightforward physiology at the neuromuscular junction. Is everyone clear on this part, folks?
Yeah.
Everybody is clear? Okay. Now, this toxin, the botulinum toxin, is encoded by the phage DNA. The phage DNA is the one that carries all the information for the botulinum toxin, and it's an AB toxin again. Just like the other toxin, we talked about the AB because this is also an exotoxin. It belongs to the AB toxin. The B part, it attaches to some of the specific receptor on the nerve motor endings. So this is the motor end, this is the motor neurons which is supplying to the muscle cell. It goes, attaches to some specific receptor on the motor cell nerve ending. The part A is the one that goes inside the cell and it becomes an active peptidase. It becomes an active peptidase, and that specific enzyme, it would degrade a vesicle protein which mediates the fusion between the vesicle membrane and the plasma membrane of the nerve cell. That specific protein is called as a snare protein. Snare is an acronym which is a long name, the snare protein which have soluble NSF attachment receptor protein. And what the heck is NSF? It is again one of the soluble, how you solubilize those NSF protein. That's why the word NSF is there. Anyhow, what I want you to know is that vesicle protein is a snare protein. Okay? So in short, this is what it does. The botulinum toxin, what it does the snare protein is degraded. Because the toxin has the peptidase or the proteolytic activity. So it chews up the snare protein, and once the snare proteins are chewed up, that vesicle will not be able to fuse. And when that vesicle is not able to fuse, so those acetylcholine will not be released at the neuromuscular junction. So botulinum toxin basically it stops the release of the neurotransmitter at the neuromuscular junction. The next picture talks a little, shows you the details of the snare protein, and there are two classes of the snare protein. The V snare. I'm not sure if anybody has heard about these snare proteins, or possibly you might have heard in your physio class if I'm not mistaken.
The one that hold the,
Huh? Have you heard about it?
the one that holds the
The vesicle.
neurotransmitters, right?
Huh?
Yeah, the ones that hold the neurotransmitters.
Yes.
So it doesn't.
That has a snare protein. Now there are two, anytime the snare protein, there are two parts of it. One is called as a V snare, and the other one is a T snare. So a V snare and the T snare, these are the two components of the snare. I won't say two components, there are two classes. There are two classes of the snare. One is a V snare and the other one is a T snare. That prefix or the small case V or the T, that represents the vesicle snare. So this one represents the vesicle snare, V-E-S-I-C-L-E, the vesicle snare, and that T stands for the target snare. Now, what the botulinum toxin, and you don't have to worry about all of these, synaptobrevin and other ones. Just remember the snare protein, because this is from a different, this is from a medical, not a medical, but a science journal, but still it gives you the idea, the take home is very clear. There is the vesicle containing this acetylcholine has the snare protein, and the muscle cell membrane has the snare protein too. These two snares, the vesicle snare and the target snare, they must interact to induce fusion and the release of the neurotransmitter. Everybody agrees? You want me to repeat it again? Mm-hmm. Sure. Thank you. So the snare proteins, because you have these two classes, the V snare and the target snare. It's like I'm the one, I'm extending the V snare, she has the T snare.
And it creates a neurotransmitter.
It doesn't create a neurotransmitter. It causes the release of the neurotransmitter because of the fusion.
Okay.
Okay, so there is a V snare. So here is the V snare. This is the T snare. And both the V snare and the T snare must interact. It's something like shaking hands before you hug each other and go further down. So this is the one. So before you start kissing or doing anything, the V snare and the T snare, they should interact. And once they interact, then that would cause the release of the neurotransmitters.
V snare plus T snare equals neurotransmitter release.
Do you see that? And what the botulinum toxin does, that's what is shown in this picture. The botulinum toxin it breaks down all those snare protein. Look at this, those snare proteins are degraded, and once the snare proteins are degraded, this vesicle is not going to come and fuse with the membrane of the muscle cells. If the vesicle don't fuse, obviously there won't be any release of the acetylcholine, and that's when you get a permanent state of the muscle cells do not get it, and you get what is called as a drooping eye, or the muscle becomes flaccid.
Doesn't that happen in stroke?
Huh?
Doesn't that happen in stroke?
You don't get the botulinum toxin causing stroke per se.
Then why do people do like Botox?
The Botox, okay, cool. What people do Botox is more like a cosmetic thing. You want to tighten your muscles, like they do for the eyelids and all. That's what they do. But it's a very risky affair. It has to be done by a person who is very well experienced, because if you put more of the Botox, it will go in the other direction.
Look like droopy.
Yes, it will make it worse, or it could have some like global effect on the body. Is that clear to you? You don't look very excited.
Is that name just a shortened version of botulinum toxin?
Which one?
Botox?
The Botox is a trademark name of the botulinum toxin. That's right. And there are many different types, so don't worry about the A, B, C, D botulism type. Don't worry about that. Well, here it shows once the botulinum toxin is present, again, the botulinum toxin will be picked up. It will be picked up, and then there's the A and the B part, but eventually the botulinum toxin will degrade all the snare protein. And because the snare proteins are disintegrated, the membrane fusion doesn't occur. No membrane fusion means no release of the acetylcholine. Yes?
So I think you said that the T snare was part of the muscle cell?
The T cell is the T1. So there is a V snare and the T snare, and the T snare will be on the muscle cell because that's a target.
On the muscle side of the
Yes.
membrane of the neuron.
Uh-huh.
So it's part of the neuron.
No,
not the
it's a
muscle.
part of the muscle. The neuron will have the V snare because of the vesicle. And they will, I'm sorry. You're correct. So the vesicle snare are here, and the target snares are on this membrane. So that's when the vesicle and the target snare will fuse and mediate the exocytosis.
Okay, what?
So the,
Yeah.
so the correction was that the T snare is actually on the neuron's membrane.
That neuron membrane
Not the muscle.
with
And
the
V
T snare
So
here,
there's not a snare on the muscle cell.
there's no snare on this one because here the acetylcholine will be picked up. I'm sorry, yes. So there's a T snare here, there's a V snare here. These two must interact so that the vesicle can now release all the acetylcholine in the synaptic cleft. Yes.
B snare, T snare gets together.
Yeah, as it's shown over here, so here. So here is the T snare, this is the V snare, and all of these are broken. So now there's no membrane fusion of the vesicle and no release of the neurotransmitters. Yes, sir. You don't look very excited. You want me to repeat it again? Anybody? You might, huh?
It's 7:30.
Oh, it's still 2 minutes somewhere. But I'm getting carried on this one. There is a vaccine. You have a vaccine against botulinum. But did you get a vaccine against botulinum?
No.
It's not given to the general population. So it's not a part of the routine vaccine because the risk of getting botulism is very, very low for the general population. That's why, although we do have an effective vaccine, it is normally not given as a part of the routine vaccination program.
Professor,
There is, yes.
so if you got that vaccine when you already had the symptoms, we'll be able to reverse this?
You know what? No, because once you have the symptom, that means you have gone too far.
Okay.
But at the same time, the incidence of botulism is not so prevalent in this part of the U.S. It's not so prevalent. But there are certain places where botulism is very common. Can you tell me where it is very common? Don't tell Texas. Don't tell New York.
Dented cans.
Huh?
Dented cans.
Where?
Dented cans, you can get botulism.
Yeah, you can, but then the canning industry is well regulated, you know, so if you're anytime going to buy some canned food and if you find that the can is puffy or something like that, don't buy that, because more than likely, because of the can, sealed can, that creates an anaerobic environment, and more than likely botulism bacteria are growing inside the can. But still, you didn't answer me where in the USA there is more incidence of botulism. Alaska? Why?
Lots of canning.
Water.
Because of the cold? So how it is with the cold? New York is equally cold. Good. Good. You know, it's very prevalent in Alaskan population.
Why?
Why? That's a good question. The reason being, Alaska folks, what they do is they smoke, what is that called? Sausages, sorry. They make sausages and they smoke sausages. So if they're not careful, sausage meat might be contaminated with the botulinum spores. Okay? Now, when they do the smoking, you know what they do is they tighten the ends. Am I correct, folks?
True.
I just go and buy it from, from traders. I don't know how to do it, but you know what my understanding is, they put it in a membranous thing and they tighten the two ends and then they slowly roast it over fire.
True.
Now, if the spores are there, there is a high concentration of nutrient because of the meat. And when they're trying to smoke it, the temperature is so fertile for the spores to germinate, and then they form the botulinum, the botulinum bacteria there. And when you eat those sausages, more than likely you might be getting a little bit more than the sausage. Okay? So you'll be getting a little bit more than the sausage, and those Clostridium botulinum will germinate. If the spores are there, then the spores will germinate, or the Clostridium botulinum will secrete the toxins, which will be absorbed and it will cause a problem. You get it, folks? So oftentimes the bacteria secretes it, and then it is absorbed, or it gets into the bloodstream, and then it is globally distributed within our body. So this is all in all about the Clostridium, the botulinum, excuse me, the tetanus toxin. Oh, I can't finish.
No, it's definitely after 7:30.
Now, our class, there is some confusion about the cell wall. What is a confusion?
The cell wall.
Oh Lord. We talked about this, the backbone. We talked about the backbone, nagnam, and so forth. And here, I'm doing it in a